Afternoon, folks. Hope everybody's doing well today. This is going to be the start of our final unit for the course, where our focus is going to be on genetics. We've now taken a look at the basic chemistry of life, proteins and carbohydrates and lipids, and seeing how they function. We've examined the structure and function of cells, including such topics as how cells manage their energy, how cells reproduce via mitosis or meiosis, and how cells move things across their membranes. But now we're going to get to the final major topic of the course, namely the question of how do cells know what to do? We need a source of information. We need a way by which our cells can store genetic data that tells them how to carry out all of their various life activities. The cell has to have a way of knowing how to build all the proteins it requires and how to carry out all the processes that it needs to execute. And so we need a chemical storage molecule, a mechanism by which the cell can have a physical structure, kind of like its own version of a hard drive, to encode all of its information. And that structure has to be something that is not only available for the cell to use, but also something that can be readily replicated and passed on to new cells during cell division. I am, of course, talking about DNA, or deoxyribonucleic acid in the longer form. DEOXYRIBONUCLEIC, deoxyribonucleic acid. Now, by the way, if you are taking this for my biology class, you might want to look on the Blackboard site and pick up a copy of the handout there. A lot of the spellings of these and other terms are going to be present there. It'll help you a lot in following along here. So DNA, or deoxyribonucleic acid, is in a lot of ways the master molecule of life. This is the chemical storage mechanism that contains, encoded within its structure, all of the information on how to make you you. Everything from the color of your eyes to the structure of your digestive enzymes. Now, your actual appearance, your phenotype. There's some terms you might have heard before: genotype versus phenotype. Genotype, G-E-N-O-T-Y-P-E. That is the genes that you have, the information encoded in your DNA. Your phenotype, P-H-E-N-O-T-Y-P-E, is your outward appearance. Now, your phenotype is a combination of genetic information and environmental influences. That your appearance is not strictly determined by your DNA, that's the main part of it, but it can be modified by a lot of environmental forces that control how those genes are expressed or how those features of the body are modified once they have been constructed. So your DNA is not the entire story on what features you end up manifesting, but it's the starting point. All of the basic structures and functions of you are encoded in that molecule. So over the course of the next couple of chapters, we're going to be exploring the chemical structure of DNA. We're going to be learning how DNA encodes information. We're going to learn how we use RNA to access that coded information and actually use it to build functional proteins in your body. And we're going to learn about heredity and how traits are passed on from one generation to the next and why certain traits would or would not be passed on. So, getting started. It's termed deoxyribonucleic acid. DNA is a nucleic acid. N-U-C-L-E-I-C, acid, nucleic acid. It's one of the four basic categories of organic molecule. Back at the beginning of this course, we talked about proteins, we talked about carbs, we talked about lipids. Now, we're going to be taking a look at the nucleic acids, the fourth major category of organic molecule. Now, nucleic acids have a couple of different functions. If you want to be technical, ATP that we talked about a while back with energy is a nucleic acid. But most of the time when you're thinking nucleic acids, think information storage molecules. Molecules whose entire function is basically for the coding and transmission of data in the form of their chemical structure. There are two major nucleic acids we talk about in living systems, DNA and RNA. They are similar in a lot of ways, but they have very different roles in our cells in terms of what they are doing. Now, I feel like a good way of starting to talk about the chemical properties and general properties of DNA is to look a little bit at the history of the molecule. to learn what we know about DNA and how we learned it. So what I'm going to be doing right now is taking you through a capsule history of DNA research. We're going to learn the names of some great scientists who are involved in this. And along the way, we're going to learn some important things about the structure of DNA and how it was all figured out. One thing I want to put up front that's really important to know about DNA. The DNA molecule is considered to be totipotent. 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 And what that means is that every cell of your body contains a complete copy of your genetic information. In other words, it's not like, okay, skin cells only have the genes for doing skin stuff and muscle cells only have the genes for doing muscle stuff. Every cell of you has all the data. I can take a sample of DNA from your bloodstream and find out information about the genetics of your liver or the structure of your skeleton, because every cell in you has all of the data. It's just that not every cell is expressing all of that information. The reason for this is quite simply because when you start out as a zygote, you start out as a fertilized egg, half your chromosomes from mom, half your chromosomes from dad. That right there is all the genetic information that you are working with. And then when that zygote begins undergoing mitosis and dividing again and again and again, making more and more cells, each of those cells is, at least initially, an identical copy of the mother cell that was the zygotes. And therefore, when they're undergoing their mitosis, they're copying all the chromosomes. There's no process by which we're sorting out, okay, this is going to become a skin cell over here, ergo we're only going to copy the parts of the chromosomes involved with skin. Now, you just get all of that stuff in there, and various regulatory molecules are going to control what is or is not expressed. We have an entire collection of genes known as Hox genes, whose entire job is to control the expression of other genes. But that's the topic for right now. So, starting at the beginning, when you hear DNA and you hear about the discovery of DNA, most people's minds immediately go to a pair of gentlemen by the name of James Watson and Francis Crick, who in the year 1953 published a series of 5 papers in the journal Nature detailing the chemical structure and function of DNA. But we're going to start our story well before these guys, and indeed the Watson and Crick story has a little wrinkle or two in it that you're going to need to know. But we're going to begin our examination way back with the discovery of the DNA molecule, which happened not in 1953, but way back in 1869. In that year, a Swiss hospital physician, a very young doctor by the name of Friedrich Miescher, F-R-I-E-D-E-R-I-C-H, Miescher, M-I-E-S-C-H-E-R, Friedrich Miescher. Friedrich Miescher was a physician in the city of Zurich in Switzerland. He was a hospital doctor, and he was doing research involving the treatment of surgical patients post-surgery, and he was studying the contents of the pus found on surgical bandages. Now, as he was doing this, Miescher discovers that there is a kind of organic molecule present there that is not one of the types that is then known. By his time, proteins, carbohydrates, and lipids have all since been documented by the new, relatively young field of biochemistry. But Miescher says, hey, I got another kind of molecule here that isn't any of those things, that it's something else. Miescher documented that whatever this new molecule was, it was always found in the nucleus of the cell. For that reason, he gave the molecule the name nuclein. N-U-C-L-E-I-N, hence the term nucleic acid. Over the next couple of years, Miescher and his students studied this nuclein in somewhat more detail. They realized that it had certain basic chemical units that we call nucleotides, but they were never able to determine what exactly it was for. They did not learn the purpose of the nucleic acids. So they kind of got forgotten about in the biological sciences for a while, since nobody was really sure what to do with them. Now, it should be understood, folks, that throughout the late 19th century, one of the big questions in biology, now that we knew what a cell was, now that we knew how the basics of cellular chemistry worked, we realized that some kind of hereditary molecule must exist, that there must be some sort of physical structure that was being passed from one cell to the next, which contained the information on how to make it do its job. So one of the big topics in late 19th and early 20th century biology was the question of what is the genetic molecule? Where in the cell is genetic information stored, and how is that done? Now, following the discovery of mitosis by more than one scientist, a couple different people independently got to it in the late 1860s, early 1870s, it was abundantly clear that during the process of mitosis, you take the chromosomes out of the nucleus, you separate them into two groups, and you put them into new cells. It was not that hard to deduce that something about the chromosomes must be involved with genetic data. So by the end of the 19th century, it was widely accepted that the chromosome was the hereditary unit. But most people got this wrong in terms of how they thought it worked. Think back to your discussion of chromosomes. What's a chromosome consist of? It consists of DNA wrapped tightly around packaging proteins called histones. Now, DNA is a fairly simple molecule. It actually only has 4 basic kinds of monomers. Proteins have 20 basic monomers, 20 different amino acids used by all living things. Therefore, proteins are a much more complex kind of molecule than DNA is. As a result, most biochemists, taking a look at the chromosome, saw DNA wound around histone proteins, found that the DNA was much less structurally complex than the protein was, and they concluded that the histone must be the part of the part of the chromosome that stored genetic data. They thought the packaging spools were the actual site of genetic information. And you can see why the logic would go that way, right? After all, genetic data is necessarily going to have to be pretty complex. It has to encode everything about you. And a protein is a much more structurally sophisticated molecule than a nucleic acid is. Therefore, the biochemists of this era thought, all right, clearly the protein is the complex part here. Therefore, when I'm looking at the chromosomes, I'm going to look at the histone and try to figure out how exactly the histone is storing genetic data. So they'd gotten the part of the cell that it was in right. They had gotten the molecular basis of heredity completely wrong. And so for decades, there was a lot of fairly fruitless investigation of the histone component of chromosomes, trying to figure out exactly how it stored data, while the DNA was generally assumed to be sort of a scaffolding. They thought that the DNA was basically a support molecule to protect the all-important genetic data found deeper inside the chromosomes. So they peeled away the DNA, looked at the histones, and needless to say, didn't find anything. Now, after a couple of decades of this, it's becoming evident that we are barking up the wrong tree, that the histones are not revealing their secrets. We are not learning anything about genetic coding from studying. And a few intrepid scientists begin saying maybe we should look elsewhere. One of those folks is a Lithuanian biochemist by the name of P.A. Levine, L-E-V-E-N-E. His full name was Phoebus Aaron Levine, P-H-O-E-B-U-S. But Levine was native of Lithuania, who was driven out of that country in the early 1900s as a consequence of anti-Jewish pogroms by Russia, which at that time had conquered Lithuania. So Levine came to New York City, got himself a position at the Rockefeller Institute in New York, which even today is a major center for biological research, and he eventually became the head of biochemistry at the Rockefeller Institutes. Levine took an especial interest in the question of heredity, and he began investigating what the hereditary molecule might be. It was evident that histones weren't working, but it was still equally evident that chromosomes seemed to be involved. So Levine said, maybe we're getting this wrong. Maybe that DNA molecule wound around the structure of my chromosome, my histone. Maybe that thing is where the genetic information is stored. So throughout the 1920s, Levine embarked on a series of investigations of the DNA molecule itself in an effort to learn whether or not DNA was in fact the hereditary molecule. And Levine made a number of very crucial discoveries. For starters, Levine realized that there were two basic forms of nucleic acids, something that Miescher and his students had suspected, which Levine named deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. So he made the DNA-RNA distinction. Levine also made the determination that the fundamental subunits of DNA, the nucleotide, had a consistent chemical structure, and he deduced the chemical structure of nucleotides. I'm going to go into a little bit of detail with this now because it actually ends up being really important to understanding how DNA works, to understand how its monomers are put together. All right, now I've got here for all of you a diagram of a DNA nucleotide. And the nucleotide is the monomer of DNA. This is the basic fundamental subunits of the DNA molecule. So here is a nucleotide, a single nucleotide. It's got several parts to it. First off, that central part, that pentagonal structure you hear in the middle, that's sometimes called a pentose sugar. The word pentose means 5-carbon sugar molecule. You can see I've numbered the carbon atoms in that molecule: 1', 2', 3', 4', and 5'. A little apostrophe next to each molecule indicates the prime symbol. You see the O there at the top. That is not a 0. That is an oxygen atom. So you've got this 5-carbon sugar molecule, this pentose sugar, that makes up the core of the nucleotide. The proper term for this molecule is deoxyribose. Hold on. Just write that down for you. So, yeah, that thing is the deoxyribose sugar, and it's from that term that we get deoxyribonucleic acid. Now, a few other things are bonded onto this sugar molecule. You can see that attached to the 5' carbon atom up at the top, you have a phosphates, PO4. Attached to the 3' carbon down at the bottom, you have a hydroxide molecule, OH. Now, those three ingredients, deoxyribose, phosphate, and hydroxide, those are the same for every nucleic acid. I mean, for every nucleotide. And by the way, I did not draw every single part of this. There are other hydrogen atoms and oxygen atoms in this ring structure. I was just drawing kind of a simplified version to show you what you need to know about it. So, deoxyribose sugar at its core, 5-carbon sugar, phosphate attached to the 5' carbon, hydroxide attached to the 3' carbon. Those things are the same for every single nucleotide. The only thing that varies from one nucleotide to the next is what is attached to the 1' carbon. And as you can see, attached to that 1' carbon, we have a molecule called a nitrogenous base. Nitrogen-based molecule, basic in solution, hence nitrogenous base. Levine discovered that there are 4 different nitrogenous bases found in DNA. And those bases are called adenine, guanine, cytosine, and thymine. So those are my 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. One of those 4 molecules is attached to the 1' carbon of every single nucleotide. Only one of them. We can therefore use the nitrogenous base as a way of identifying the nucleotide sequence. You can say, okay, this is an adenine nucleotide, or this is a cytosine nucleotide. Also, you will find that when you write out genetic codes, that you can write them looking something like that. You can write them as a string of letters, A, G, T, and C, which basically tell you, okay, the first nucleotide in this molecule is an adenine nucleotide. Then it's followed by a guanine nucleotide, then a thymine nucleotide, then another adenine, and so forth. So we can basically use these 4 units as kind of a genetic alphabet. We can spell out a DNA sequence by using the nitrogenous base attached to each nucleotide by way of identifying. So we'll talk about an adenine nucleotide or a guanine nucleotide or whatever. In terms of those molecules, I'm not going to draw their full structure, but adenine and guanine are both examples of what chemists call purines. A purine is a molecule, a nitrogenous molecule with a small single ring chemical structure, one ring of nitrogen and oxygen and hydrogen. So adenine and guanine are both purines. Cytosine and thymine are both pyrimidines. And if you can't see my diagram here, Pyrimidines, purines is Purines, adenine, Adenine, guanine, Guanine, cytosine, Cytosine, and thymine, Thymine. But yeah, so anyway, so purines, adenine and guanine are purines. They have a small single ring chemical structure. Cytosine and thymine are pyrimidines. They have a larger double ring chemical structure consisting of two interlocked nitrogenous rings. So that is our nucleotide. Our nucleotide consists of deoxyribose sugar, 5-carbon sugar, phosphate attached to the 5' carbon, hydroxide attached to the 3' carbon, and any one of the 4 nitrogenous bases, adenine, guanine, cytosine, or thymine, attached to the 1' carbon. So that's our basic nucleotide structure. Now, these things do not vary from one nucleotide to the next. The only thing that's variable here again is A, G, C, or T. That's the basic setup. Now, Levine discovered all these parts of the nucleotide. He also discovered how nucleotides bond to one another to form a polymer. Levine discovered that the way that nucleotides join to each other is that the phosphate group of one nucleotide bonds to the hydroxide of the preceding nucleotide. In other words, the 5' phosphate on this nucleotide connects to the 3' hydroxide on that one. So you always link nucleotides together vertically in this way by bonding 5' phosphate to the preceding nucleotide's 3' hydroxide. It's a covalent bond between the two of them. We give it a special name. We call it a phosphodiester bond, PHOSPHODIESTER. So a phosphodiester bond. And you can keep on the chain going. Here's another hydroxide there. We could take another one down here, another nucleotide, link its phosphate onto there, form another phosphodiester bond, and keep on building them vertically in that direction. You always assemble DNA in that way, linking 5' phosphate to 3' hydroxide. For that reason, we often say that the DNA molecule has an inherent directionality, that DNA is always built in the 5' to 3' direction. Keep that in mind. That is an important detail. DNA is always assembled 5' to 3'. That is 5' phosphate of one nucleotide links with 3' hydroxide of the preceding one. All right, so Levine learned all these crucial facts. He learned the difference between DNA and RNA. He discovered the nitrogenous bases and their chemical structure of the 4 nucleotides in DNA. He learned how nucleotides bond to each other to form a polymer. A lot of really important basic information about the chemical structure of the DNA molecule. Having done all that, Levine comes to the conclusion that DNA is not the genetic molecule. His thinking was looking at the molecule. He said, for starters, it's too simple. It only has 4 basic letters in the genetic alphabet. He said there is not enough complexity here to encode for all the features of a biological organism. Secondly, Levine said DNA is a repetitive sequence. It's just the same letters over and over and over again repeating, and therefore, in the absence of that complexity, it can't possibly be encoding the highly varied information needed to build a living thing. Unfortunately for all of science and for Levine's personal career, he had the misfortune of having studied a sequence of DNA that happened to be repetitive. In other words, he was looking at a specific bit of DNA that happens to have a repetitive structure, and therefore was misled into thinking the whole molecule was like that. And so, on the basis of that supposed simplicity, Levine rejected the idea that DNA was our genetic molecule, and he recommended we go back to studying the proteins. Now, that said, he learned some really crucial stuff about how the DNA molecule was put together. And the information Levine had garnered from all this was going to remain important and be crucial to what other things people would figure out later. Now, throughout the 1930s, DNA research does not make a lot of headway. People are, again, looking at histones, trying to figure out the best way to make them work and figure out how they store genetic data. And needless to say, nobody was getting anywhere with this. There were one or two scientists who suggested that DNA might have more going for it, but their research didn't really get anywhere. Now, this all is until the 1940s. In the 1940s, we're going to have a big breakthrough in genetics made by a rather unlikely individual, a bacteriologist, a Canadian bacteriologist by the name of Oswald Avery. Now, if you are picturing in your mind a man named Oswald Avery, you are almost certainly correct. Little tiny bald guy, little spectacles, short little fella, shuffles quietly up to his podium, gives his papers, mild-mannered, soft-spoken, most brilliant bacteriologist on Earth at the time in all probability. And Oswald Avery was a particular expert on bacterial pneumonia. Now, Avery was studying different strains of pneumonia. He was not setting out to learn about genetics. That wasn't his goal here. This was an incidental thing that he's going to learn along the way. He is studying different strains of pneumonia bacteria, trying to figure out why some pneumonia bacteria are more lethal than others. Now, he does his experiments with mice, and he's got a couple different strains of pneumonia. He's got one strain of pneumonia that is particularly lethal. The mice almost always die when they get it. And one strain of pneumonia that is a lot less lethal, that the mice get sick for a while, but they generally recover. Now, Avery did an experiment in which he heat-treated the lethal pneumonia bacteria, causing them to lyse, their cells ruptured and broke, and then he mixed up live weak pneumonia bacteria with the dead lethal ones. And he discovered that something happened that caused his milder pneumonia to become lethal after he did that. So he surmised correctly that there must be something in the lethal pneumonia that was being sucked up and taken in by the less lethal ones, which caused them to become more dangerous. And so he set out to find out what this transforming principle, as he called it, would be. Now, Avery took a look at the biochemistry here, and he noted that the bacteria were indeed absorbing some of the molecules taken up by, released by the death of their more lethal counterparts. Avery realized that there were three possible things that they could be taking in that might be causing the transforming property. He realized it could be the DNA, it could be the RNA, or it could be the proteins. Now, those of you who know a little bit about cells already have probably already figured out what was happening here, that our lethal pneumonia bacteria contained plasmids, which made them more lethal. Remember, plasmids are little tiny rings of DNA. You might call them bonus genes that only contain one or two genes each, and as a result, give the organism, the bacterium, capabilities that otherwise wouldn't have had. Plasmids aren't necessary for the survival of the bacterium, but they can help it out. What was really happening was that our weaker pneumonia bacteria were absorbing plasmids released by the death of the stronger lethal varieties, and because they got that plasmid, they gained that lethal quality. They gained that genetic feature. Horizontal gene transfer. You've heard about this before. Now, Avery doesn't know this yet. He doesn't know what a plasmid is. All he knows is that DNA, RNA, and proteins are all available as possible things being taken in by the less lethal pneumonia from the more lethal ones. So Avery devises a really clever experiment to figure out which part is the transforming principle. He repeats the exercise. He heat treats the lethal pneumonia bacteria, a sample of them, and then he infects mice. He heat treats it, but then before he mixes in the less lethal pneumonia, he divides his sample into three parts. One part of that sample he adds a DNA digesting enzyme to that breaks down DNA. One of those samples he adds an RNA digesting enzyme to that breaks down the RNA presence. And the third one he adds a protein digesting enzyme to it that breaks down the protein. So now he's got three samples of the lethal bacteria, one with digested DNA, one with digested RNA, one with digested protein. Now he mixes in the less lethal bacteria into each of those 3 samples individually, lets them percolate around and absorb stuff in there, and then he infects mice with them to see what happens. Avery discovers that the sample in which the DNA had been digested, the mice did not get sicker. In other words, the bacteria had not become more lethal. But in the samples where the RNA and the proteins were digested and the DNA was left intact, the bacteria became more lethal. On that basis, Avery concluded that the DNA must be the transforming principle, that DNA was the thing that was being picked up by the less lethal bacteria, making them more lethal. And he concluded, therefore, that DNA must be the genetic molecule. It was an incredible experiment, and it was one that by all rights he probably should have gotten a Nobel Prize for. But the sad part about Avery's research is that he was literally the only person on Earth at the time who knew enough about pneumonia to even attempt to replicate the experiment. In other words, no other scientists were able to independently verify Avery's results. As such, his results kind of languished. And the simple fact of the matter is there was also not that much crossover in the genetic literature, the scientific literature, between people doing bacteriology and people doing genetics. And as a result, Avery's research, which was published in bacteriological journals, didn't get the attention of a lot of genetics researchers. And as such, his brilliantly important finding was largely ignored and did not really end up getting any traction. But I like to give Oswald Avery his props. Now, this isn't going to last for long. Word of Avery's discovery is going to percolate out, and it's becoming abundantly clear that proteins are not giving us what we need. Now, in 1951, 2 big things are going to happen in genetics research. One of them is going to involve an experiment involving an American scientist by the name of Alfred Hershey and his young lab assistant, a woman by the name of Martha Chase, one of the relatively few women in the field of biochemistry at the time. The Hershey-Chase experiment is often considered one of the most significant medical and scientific experiments of the 20th century. Now, Hershey and Chase were studying the problem of what was the genetic molecule, but they were coming at it from a different standpoint. They were looking at viruses. Now, viruses are on everybody's mind a lot these days, and I'm going to do a whole lesson on them at some point. But what we need to know about a virus right now is a virus is not a living thing. A virus is not a cell. A virus is a small bit of genetic information, either DNA or RNA. Some viruses are DNA viruses. Some are RNA viruses. The coronavirus everybody's concerned about right now is an RNA virus. But either way, you've got the genetic molecule, either DNA or RNA, wrapped up in a coating of protein called the capsid. And the capsid is going to have features like spikes or other things on its surface that allow it to penetrate and attack cells. Now, a virus is basically a genetic hijacker. Viruses do not have any ability to reproduce on their own. What they can do is co-opt and take over a living cell, transform that cell into a virus-producing factory, and thereby make more viruses indirectly. Talk more about how that happens later. But the Hershey-Chase experiment was meant to evaluate this and to try to find out which part of the virus contained its genetic information. Because viruses are so much simpler than cells, all they have is one layer of external protein and then their genetic data, there are only 2 real parts to concern themselves with. So Avery and Chase set up a really clever exercise to try this out. They knew that at a biochemical level, proteins contain sulfur, but they do not contain phosphorus. They also knew that biochemically speaking, DNA contains phosphorus in its phosphates, but it never contains sulfur. As a result, they're like, hey, we can use that fact to label the different parts of the virus. So what they do is they culture themselves some viruses in a petri dish, but they mix in radioactive isotopes of both sulfur and phosphorus. The viruses incorporate those radioisotopes into their own chemical structure. What we have now done is what's sometimes called radiolabeling. We have radiolabeled the DNA part of the virus with radioactive phosphates, and we've radiolabeled the protein part of the virus with radioactive sulfur. And we can track the radiation given off by those things in order to find out where that part of the virus is gone. Having done that, Hershey and Chase, excuse me, exposed their sample organisms, which were actually tobacco plants. They used a plant called, they used a tobacco plant, and they used a virus called tobacco mosaic virus, which was afflicting a lot of tobacco crops in the 1940s and 50s, and therefore was a major research subject. So they did their modifications on tobacco mosaic virus, and then they exposed tobacco plants to it to find out what happened. What they discovered was the basic mechanism of viral infection. Namely, what happens is the capsid of the virus, the protein part of the virus, attaches to the outside of the membrane of the affected cell, and then it injects its DNA into the cell. The DNA is the only part of the virus that enters the target cell. Once it's in there, it co-opts the cell and forces the cell to begin manufacturing more viruses. So Hershey and Chase were able to determine this because they could see that the radioactive phosphates ended up inside their target cells, whereas the radioactive sulfur stayed on the outside. In other words, the only part of the virus that was getting into the tobacco cells was the DNA, while the protein capsule remained on the exterior surface. In this way, Hershey and Chase determined quite conclusively that DNA is the genetic molecule. And along the way, they also learned the vital medically important fact of how viruses infect cells. Eventually, Alfred Hershey would end up getting a Nobel Prize for this experiment. Martha Chase was not a co-recipient of that prize. She had completely left science by that point in time. In fact, within 10 years of the Hershey-Chase experiment, Martha Chase had completely left any kind of scientific work. She had become a housewife. She died a few years ago of early onset Alzheimer's, and one of my pet projects I'd like to get to at some point is to investigate her life story and find out why she left the sciences after engaging in such a crucial experiment, and well before her mentor got that recognition. But I digress. So, 1951, we know now for sure, and people involved in genetics know for sure, that DNA is the genetic molecule. Now, the other big thing that happens this year is a bit of research by an Austrian biochemist by the name of Erwin Chargaff, E-R-W-I-N, Chargaff, C-H-A-R-G-A-F-F. Now, Erwin Chargaff was kind of a colorful figure in the history of sciences. He was a popular science writer. He tried to write science in ways that were accessible to ordinary people. He was a very gifted public speaker. He gave lectures on science for the sake of people who didn't understand a lot about it. You know, science as lay people who weren't scientists could understand it. Chargaff was also very handsome, debonair, dark-haired, dark-eyed, tall, handsome kind of guy. A little bit full of himself. He gave out signed headshots of himself at his public lectures. Actually, if you Google Erwin Chargaff and you go on Google image search, one of the first things you will see come up is one of his headshots, you know, with his movie star kind of looks. Now, Chargaff, despite his kind of, you know, playboy attitude and his, you know, popular science focus, is a really good biochemist, and he is trying to crack the problem of DNA as well. At this point, anybody who's anybody in the history of, in the genetics field is working hard to figure out DNA. We know DNA is the genetic molecule at this point, but we haven't worked out how it does its thing. We have not worked out the chemical structure of DNA that it uses to encode information. That is as yet a mystery. So, Chargaff's one of many people trying to puzzle this out. He does not solve the puzzle, but he does learn a couple of very important things about DNA. One of the things Chargaff figures out is that DNA is not a repetitive sequence, that the DNA molecule is not, as Levine thought, just the same sequence of nucleotides over and over. Chargaff determines that it's the same in every single organism. Sorry, I misspoke. What Chargaff found is that DNA is different in every single organism. No two organisms have the exact same sequence. This made it a lot more credible as a genetic molecule. The other big thing Chargaff found was he discovered a pattern in DNA. But although the molecule was different in every organism, he did find a pattern in the nitrogenous bases. Namely, he found that no matter what organism's DNA he looked at, that the amount of adenine, the A nucleotide, present in the DNA was always equal to the amount of thymine, the T nucleotide. He also learned that the amount of the guanine nucleotide, G, was always equal to the amount of the cytosine nucleotide, C. No matter how much of each of them was present in any given DNA molecule or how those molecules were arranged, the ratios were consistent. A always equaled T, G always equaled C. He didn't know why, but he did correctly surmise that this had something to do with the structure of the molecule. Now this gets us to 1953, when the race is on among geneticists all over the world, trying to figure out the structure of the DNA molecule. People in America are doing it. The famous American biochemist Linus Pauling, who's famous for figuring out that vitamin C is good for helping you when you're sick. We've since learned it's actually not quite as simple as that. Linus Pauling is working on it in the US. A number of people are working on it in Great Britain and France. This is the hot topic, and everybody knows that whoever works out the structure of DNA is going to be initiating a new scientific revolution. This is the future of biology. And so that brings us around at last to names you probably heard before. Well, I know you heard them before because I mentioned them at the beginning of this lecture. An American scientist by the name of James Dewey Watson and his British colleague Francis Henry Compton Crick. Watson and Crick. C-R-I-C-K. Now, Watson and Crick, those names are virtually synonymous with DNA. The two young men at this time, they're both young scientists, they're fresh out of school postdocs, they're working at the Cavendish Laboratory in London, which is a major biochemical laboratory still active today. Now, I should point out right now, they're assigned to the same lab because they're working on a similar project, and they get to talking, and they discover both of them have a mutual interest in the chemical structure of DNA. And they decide that they want to try to crack the nut. They want to figure out the structure of the genetic molecule. And so they throw their hats into the ring as part of this race. This was not what they were supposed to be doing. Watson and Crick were not, in fact, assigned to be trying to figure out the structure of DNA. Their research was supposed to be on tobacco mosaic virus. They were supposed to be doing virus research. But because they were researching tobacco mosaic virus, they were very familiar with Alfred Hershey and Martha Chase's work because their project had been done on tobacco mosaic virus. And therefore, that got them interested in the DNA question, and they said, let's try to figure it out. So Watson and Crick begin spending a lot of time and a lot of Cavendish lab resources, incidentally, trying to deduce the structure of this molecule. They build a whole bunch of three-dimensional models. For a while, they think that the nitrogenous bases are on the outside of the DNA molecule. For a while, they think it might be a three-stranded molecule. They're building a whole bunch of different models. And they learn some really important chemical things about DNA and its basic structures and how it's put together. But they have not yet figured out the overall structure of the molecule. That is still eluding them. They work out a lot of important details about it. I'm not going to go into the fiddly biochemistry of the specific things they're finding. But they have not yet figured it out. And in fact, they are told by their boss at the Cavendish Laboratory, guys, this DNA thing is not your project. Cut it out. Focus on what you're supposed to be doing. But they didn't stop. They continued pursuing the project quietly in the background. Now, there is somebody in London whose job it is to be figuring this out. Across town at King's College in London, there is a biochemist by the name of Rosalind Franklin, R-O-S-A-L-I-N-D, Franklin. Rosalind Franklin is one of the very few women who has made a credible reputation for herself in the field of biochemistry. She and Martha Chase are pretty lonely figures in that regard for women in the hard sciences. Quite frankly, science in the 1950s was still a boys' club. And although scientists like Marie Curie had made great strides for women getting into science, the simple fact of the matter is that women are not taken seriously as scientists. It's extremely hard as a woman scientist to be taken seriously. It's hard to get into scientific or medical programs. It's difficult to get in control of your own research, even if you do successfully get a science education. Male scientists just don't think of women scientists as being equals. Now, Rosalind Franklin has managed to carve out a name for herself. As a very gifted biochemist, she first did her research in coal. Actually, some of her coal research is still cited to this day. But then she later moved on to an interest in genetics. And specifically, Rosalind Franklin was an expert with a technology called X-ray diffraction, D-I-F-F-R-A-C-T-I-O-N, X-ray diffraction. And this is a technique in which you use X-rays to photograph things at the molecular scale. You cannot photograph a molecule with a standard light-based photograph because individual molecules are smaller than waves of light, and therefore you can't bounce a wave of light off of them to take a picture. But X-rays have a much tinier wavelength. They are much smaller than individual molecules. And therefore, you can use this technology to basically embed a molecule in a crystal, shine X-rays through it, and assemble an image of the molecule in that way. And Franklin is using this technology in an effort to get an image of the DNA molecule. She gives some papers about her research. She gives a couple of talks about it. Actually, Watson and Crick attend one of her talks and are quite impressed by the work that she's doing. Now, Franklin has not yet gotten there either, but she's close. She has nearly got the structure of DNA worked out. She's this close. But there's a fellow in her lab by the name of Maurice Wilkins, W-I-L-K-I-N-S. Maurice Wilkins is a good enough scientist that he's at the King's College Laboratory, but he is quite frankly not the guy who's going to be making blinding, amazing discoveries all on his own. And Maurice Wilkins badly wants a piece of that DNA pie. He knows what this means. He knows that the future of science is in DNA, and he knows that whoever works out that structure is going to become immortal. Wilkins badly wants on Franklin's project, and he pressures her again and again to let him participate in her research. But Wilkins, like so many scientists of his day, was a bit of a chauvinist who did not really take women seriously. He was very patronizing and condescending to Rosalind Franklin, and Franklin knew very well that if she let Wilkins in on her project, he was going to totally co-opt her research. And so she rebuffed his efforts to get involved. Wilkins got pissed about this. Without Franklin's knowledge, he goes into her laboratory. He removes X-ray diffraction images she had taken of the DNA molecule, including the famous Picture 51, which you can actually Google and find online, and takes these images, puts them in an envelope, passes them along to Watson and Crick and says, "Hey boys, I know you're still working on that DNA project. Here's a little something that might help you out." Watson and Crick, with the combination of their previous work and Franklin's images, make the breakthrough. Putting that stuff together, this is the last piece of the puzzle that they need. And so, May of 1953, 5 papers are published in the journal Nature detailing the chemical structure of the DNA molecule. Those 5 papers initiate a revolution in science. Biology has never been the same since then. DNA is the single most important scientific discovery, I would argue, of the 20th century, or the structure of DNA. It has enabled virtually everything we now know about the biosciences and medicine to move forward. It's clarified the workings of evolution. It has clarified how genetic disorders work. It has basically changed the entire way we look at the natural world. Most people do not know that there are 3 listed authors on those papers published in 1953. James Watson, Francis Crick, and Maurice Wilkins. Rosalind Franklin got not so much as a footnote acknowledging her contribution. Because Rosalind Franklin never knew. She literally never found out what had happened. Looking at Rosalind Franklin's research papers now, we've determined that she herself was about a month or 2 out from the big discovery, that had Watson and Crick not gotten her images when they did, it is highly probable that by the end of that summer, Rosalind Franklin would have been the sole author of a paper on the structure of DNA, and hers would be the only name in the story that you ever heard. But Franklin never knew her images had been stolen. She, in fact, wrote a review of Watson and Crick's research, praising them for their clever discoveries. Franklin continued her own work, but her career did not last a lot longer. By the time that Watson and Crick stole her images, she was already sick. Her frequent work with X-rays had given her breast cancer. And, sorry, not breast cancer, ovarian cancer, my bad. And she succumbed to ovarian cancer 5 years later when she was only 38 years old. So Rosalind Franklin's career was cut tragically short as basically a casualty of the dangers of radiation from her own research. This is before we had good shielding and so forth. In 1963, James Watson, Francis Crick, and Maurice Wilkins shared the Nobel Prize in Medicine for the discovery of the structure of DNA. They have been lionized in the sciences since then for their work. None of them saw fit to even mention Rosalind Franklin at the Nobel Prize award ceremony. Now, Wilkins, for his part, never went on to do anything else of real significance in biology. James Watson went on to a very illustrious scientific career from that point onward. He taught at Harvard for many years. He worked out the central dogma of biology, which we'll discuss in a future chapter here. And in the early 20th century, he teamed up with J. Craig Venter. You might be aware of this already. And in the early 21st century, in the first couple years of the 21st century, he and Venter completed the Human Genome Project, the effort to sequence the entirety of the human genome and learn the locations of every gene in human DNA. Francis Crick went on to considerable research of his own in biochemistry. He worked out the structure of the codon as the basic unit of genetic information, as well as learning about some things involving RNA. Fortunately for Rosalind Franklin, her story was known by someone, and that someone was her young lab assistant, a man by the name of Raymond Gosling. Gosling went out of his way to make sure that Franklin's story got told, that people knew what had happened, that they were aware of her vital contribution to the science of genetics and exactly how that work had been co-opted. Thanks to Gosling, we know the story. Now, nearly everybody involved in this whole sordid affair is dead now. Francis Crick passed away back in the early 2000s. Maurice Wilkins passed away about the same time. Both of them, before they died, admitted to what they had done wrong, fessed up to this, expressed great regret for their crimes against Rosalind Franklin. Francis Crick, before he died, said, "Let none of you speak of myself and James Watson in the same breath in which you do not mention Rosalind Franklin." The only person involved in this whole affair who is still alive is James Watson himself. Watson has never admitted to anything wrong. Despite Crick and Wilkins both having said what actually happened, James Watson has refused to own up to the theft of Franklin's data. He wrote about her in his book, The Double Helix, on the basic history and structure of DNA, but his writing about her is very patronizing. He called her Dear Rosie, which was a nickname she hated, talked about her being a clever girl, but not someone who was going to make big discoveries all by herself. If you're getting an impression, James Watson is, well, I'm not going to pull any punches here. James Watson lost his position at the Cold Springs Harbor Research Laboratory in New York several years back, whenever he gave an interview on the anniversary of his and Crick's research, talking about the future of genetic science. And in that interview, he said that he believed that the persistent political and economic problems facing the continent of Africa are a consequence of genetic inferiority in the intelligence of native Africans. He followed that particular gem up by saying, any of you who have ever had an African-American lab assistant know what I'm talking about. Needless to say, that particular sentiment, coming from a man whose very research proved that that's not how this works, is especially monstrous. And thankfully, the scientific community said, no, we are not putting up with that bullshit. Watson was pretty much blackballed from research thereafter. He was forced to resign his position at the Cold Springs Harbor Research Laboratory in New York about a month after that interview was published. And Watson has since spent his time in this growly, angry old man retirement where he sends out nasty tweets from his basement talking about how he's working on this secret genetic project that's going to show us all, apparently. But he's pretty much a non-entity in science now. And quite frankly, as far as I'm concerned, good riddance. Because if you are taking a lesson from all of this, it is that the use of science in the service of prejudice and falsehood is pretty much the cardinal sin of our profession. Rosalind Franklin deserved far better than she got. It makes me very happy that in these days of modern science, her name gets passed around quite frequently. I'm hoping some of you in here heard of her before you heard me mention her today. That there's a wonderful biography of her, by the way, called Rosalind Franklin, The Dark Lady of DNA by Brenda Maddox. I strongly recommend it. It tells her story in great detail, much better than I've given you right here. So, moral of the story, folks, listen to what Crick said before he died. If you mention him and Watson, mention Rosalind Franklin in the same breath. Now, it is true that Watson and Crick did some really important stuff, but the simple fact of the matter is they would not have finished the story without Rosalind Franklin. It was the combination of their work that gave us our understanding of the structure of DNA. And therefore, although Watson and Crick were frankly thieves who were plagiarizing their work, they were committing the cardinal sin of science. we must acknowledge they did some important work on their own, but we also must acknowledge Rosalind Franklin's contribution. It was just as important as theirs. It was what gave them the final piece of the puzzle. She had solved what they had not, and we could not have solved the issue without her. So if you want to be fully honest in your presentation of this, let's say Franklin, Watson, and Crick, because those are the people involved, and frankly, I'm going to put Franklin first. So that is our capsule history of the science of DNA. And my charge to any of you who are students of science listening to all of this is that we are not done with this nonsense. James Watson's persistent racism and misogyny just goes to show us that this is a fight we're still fighting. And I hate to say it, but women in the sciences still have an uphill climb, because there are still a lot of grunting, Cro-Magnon old men scientists who somehow think that being a woman means you have no place in a laboratory. And my charge to all of you listening to this is, let's stop that bullshit right here and right now. If you're a woman listening to this, don't put up with that crap. I'm sorry, you're going to probably have to deal with it at some point if you already haven't. But take a stand. And to the men listening to this, use your privilege to shut this down. If all of us stop doing this nonsense, if all of us agree that we cannot move forward with the study of the things important in the understanding of the natural world without the contributions of half of the human race, we can choose to stop this nonsense. We can choose to not be bigots. Check your privilege. If you see this bullshit happening, stop it and don't commit it yourselves. You do that, we can get over this problem. We can all learn a lot more. All right, folks, we're in a little bit long here, but I think it was an important overage to get into. Next time we get together, we're going to learn more about the structure of DNA itself. What is this molecule all about? What did Franklin, Watson, and Crick discover? And what can we learn from it to learn how DNA codes and decodes your genetic information? Thank you for your attention, folks. Hope you all have a great rest of the day.